Hi, thanks for sticking around. I'm going to talk today primarily about two studies done on the exact same population aimed at the same question, employing a fundamentally different approach, and they give two very different answers. So that question is what maintains sexual reproduction, specifically given the twofold cost of sex. So females pay what's called the twofold cost of sex, and that they only, when they reproduce sexually, they only transmit half their genome to their progeny, whereas an asexual female would transmit her entire genome to her progeny. So what prevents an asexual female from invading a sexual population? And one hypothesis for this is that by having males in the population and strong sexual selection on males, males can be used to purge the mutations population, purge the population's mutation load, increasing population mean fitness or population viability. And in order for this to happen, there are two primary criteria. So the mutations that we're talking about should be selected against more strongly in males than in females, and they should have what's called mutational pleiotropy. So they should, those that are selected against in males that are detrimental to male lifetime competitive reproductive success should also be detrimental to population productivity, which is a largely female-driven trait, so female productivity. We use a seed beetle called Aspergillus maculatus. They lay their eggs on beans. They develop inside of the bean. They emerge as reproductively mature adults. It makes them nice to work with in the lab. They don't need food or water as adults. They just reproduce. So our fitness assays in the lab are quite relevant to what they experience in nature. We took a wild population from Loma, Togo in Africa, and we cut it up into isofemale lines back in 2012. One of the original characterizations of that population revealed that it was dominated by sexually antagonistic genetic variation for fitness. So we have genotypes with high female fitness that have low male fitness and vice versa. And the first study that we did that was aimed at the question I posed in the beginning used induced mutations by ionizing radiation to see how they're selected in males and in females. And so we had an irradiated and a control group. We had mutations coming in through females or through males. We had male and female controls, which are the same, actually, so they're pooled in the results. So mutations induced in F0 generation. In the F1 generation, we employed a middle-class neighborhood breeding design so that those mutations were not selected against in this intermediate generation. We also used the offspring produced in this F1 generation as our estimate of population productivity. So these were just single pair matings. And then from those offspring that pair's productivity, we took 2 males and 2 females from each of those families, and we conducted competitive lifetime reproductive success assays. So we have a focal individual competing against a sterile reference competitor of the same sex for access to 2 members of the opposite sex. And so in the case of males, this includes mating and sperm competition for the lifetime of the assay, the lifetime of that individual. And we found whether you split this up between mutations that were induced in grandfathers or grandmothers, or whether you look at each genetic background differently, independently, we found consistently stronger selection in males against this induced mutation load relative to females. In fact, it wasn't there for females at all. So strong sexual selection against this mutation load in males, but not in females. This was analyzed by frequentist and Bayesian approaches. They were both congruent. And so then, does this mutation load that was selected against in males have detriments to population productivity? And we find yes, that whether they were induced in grandfathers or in grandmothers, there's this positive correlation between our F1 productivity estimates and our male competitive lifetime reproductive success in the F2 generation. These 2 ellipses here, this gray and this black one, are positive correlations. Whereas in the controls, this is just a flat relationship here, so there's no relationship. There's no relationship based on the standing genetic variation between male competitive lifetime reproductive success and population productivity. So we found that there was strong sexual selection in males against the mutation load that we induced that reduced offspring production in seed beetles. So this is in line with the hypothesis that there's a value to males in a population. This poses some benefits to sexual reproduction or mitigates some of the cost of the twofold cost of sex to females. I took those isofemale lines and I inbred them for 10 generations further. I actually inbred hundreds of them, and we ended up keeping 16. That was manageable. So this is how those 16 were distributed about the original intersectional genetic correlation. So it's a pretty random representation of genetic origins. I performed a diallel cross among those 16 inbred lines. So this is a sex-specific factorial cross, which gives us 256 cross types. And from the offspring of those crosses, then I did the same lifetime competitive reproductive success assays that I talked about earlier for males and for females separately. And we get two things out of here. So along the diagonal, these aren't really crosses per se. These are within line single pair matings. So this is their inbred fitness, if you will. And on the off diagonals, then we've got their outbred fitness. So if we sum this across the sire and dam contributions, you've got a sort of global estimate of the outbred fitness of each of these inbred lines. It was a big data set. So this is just to give you a sense of what we're looking at here. So on the left, this is the on the diagonal, and this is on the off diagonal on the right. And so their inbred fitness, there was a positive intersectional genetic correlation here, which makes sense because what you're doing is inbreeding them to the point where you've revealed this recessive deleterious mutation load, which tends to be detrimental to both males and females. So it's a positive intersectional genetic correlation. There's no relationship here, so each of these inbred lines responds differently to being outbred, and that's what we're going to look at right now. Okay, so their response to being outbred is going to be their heterozygous fitness, and we're going to look at how that's related to their homozygous fitness. So I'm going to call this mutation load. This is variation in mutation load, and this is how they respond to being outcrossed. So this is how they respond to having that deleterious recessive mutation load covered up by dominant alleles. And if that outbred fitness reveals variation in mutation load, then there should be some relationship here. It should be greater than 0, but this slope should actually be less than 1. And that's because you would expect that individuals down here on this end with low homozygous fitness have a lot of mutation load, so they benefit more from having that covered up by dominant alleles. Whereas individuals out here with high homozygous fitness, they have less mutation load, so they benefit less from having that mutation load covered up by dominant alleles. So this should be significantly between 0 and 1, these slopes. So they're going to look like this. I'm going to plot the slopes as red and gray lines as confidence intervals for those slopes. So now we're doing a question, we're doing a study on the same question, but we're looking at the standing genetic variation rather than previously we were looking at induced mutations. So based on the standing genetic variation, we find that variation outbred, variation in male fitness reveals variation in inbred male fitness. So this is to say that there's sexual selection in males against the mutation load on male fitness. And that's not the case for females. So there's no relationship here. So this is congruent with the first result from the first study, and that there's strong sexual selection against this standing mutation load in males, but that's not the case in females. Okay, so then the big question here regarding the mutational pleiotropy is, does outbred male fitness reveal the mutation load on female fitness? Or in other words, does outbred male fitness reveal mutation load on population viability? Because population viability is going to be a female-driven process. And there is no relationship there either. So what's going on here with regard to the standing genetic mutation load? Is there some kind of a private male genome? Variation in male fitness is revealing the male-specific mutation load, but not the mutation load that's driven by females. And so there's not going to be any value to males here. This isn't going to mitigate the twofold cost of sex to females. So we can get at the question of, is there a private male genome a little bit more by cutting this data set up a little bit differently. So remember, I have sire and dam contributions here. So I was averaging cross sire and dam contributions. Now I can split that up and say, what about the outbred male fitness that comes from their fathers, or the outbred male fitness that comes from their mothers? So that which they inherit from their own sex or from the other sex. So if we look at sire inherited male fitness, it's the same story. Outbred male fitness is revealing the mutation load on male fitness, just like earlier, but not the mutation load on population viability. And if we look at that outbred male fitness that's inherited through their mothers, so the group that data set by dams rather than by sires, that relationship falls apart. So now outbred male fitness doesn't even reveal the mutation load on male fitness. When you ask if outbred male fitness reveals the mutation load on population viability, it's such a poor relationship they can't even estimate the confidence limits for this slope. These are range access regressions. So, to conclude here, then we've got strong sexual selection in males, but only against an induced mutation load, so only against novel mutations. Whereas the standing mutation load that's sexually selected against in males seems to have male-specific effects. So, so what's going on here? This is similar actually to a result that we found in another study on the same population, where the isofemale lines that have high male fitness and also have male-like suites of life history traits have lower population growth rates. So there's high male fitness is dragging down population growth rate. So some people might say, okay, well, you probably found this result because your population is sexually antagonistic to begin with. It's dominated by sexually antagonistic genetic variation. But I think the opposite is probably the case. I think that this process, as what is making the population have predominantly sexually antagonistic genetic variation for fitness. And this is not a new idea. This was pointed out by Karl and Clark and others, that when there are new mutations that are coming in and they have sexually concordant effects, that they're going to be rapidly fixed or eliminated, which leaves you with mutations that have sex-specific and sexually antagonistic effects. So can males serve as a purifying force? Yes, I think that they can, which is exactly why you won't see that if you're looking at the standing